इसमें कितने लोग ज्वाइन हुए ये भी पता चलेगा। यहाँ पे चैट्स आती है ना। हाँ हाँ। सॉरी सॉरी। Very good morning to all participants.
Good morning.
And our today's speaker, Professor C. Prabha, Professor in Head Department of Biochemistry, MS University of Baroda. And today with us we have the Assistant Dean of Center Research and Development, Dr. Ashish Rami. He is also the head of RAI Business School. So I, Dr. Sandesh Chibbar, Head School of Life Science, welcome you all for the live webinar on protein engineering. So before we move on to today's talk, let me brief you about the School of Life Science. RAI University, we offer bachelor, master and doctorate program in biotechnology and microbiology. Besides, we also running Bachelor of Technology program in Biotechnology, Environment and Food Process and Technology. From 2020 onwards, we have introduced dual degree program both in biotechnology and microbiology. We, the School of Life Science, working on the vision to get recognized at regional, national and international level in research, undergraduate and postgraduate education by creating allied knowledge and technology in the field of life science as a multidisciplinary field. And our mission is to engage in cutting edge research, education, industry oriented skills development in students, and offer help to the community with an aim to discover, develop, and disseminate knowledge in the biological science. With this, I would like to welcome our today's speaker, Professor C. Ratna Prabha. I take this opportunity to introduce our today's speaker, Professor C. Ratna Prabha. Professor Prabha did her PhD from IIT Bombay. Professor Prabha has been awarded with Brain Korea 21 Fellowship, for which she moved to Gwon Institute of Science and Technology, South Korea, for her postdoctoral research, and then she moved to Cambridge University, UK, for her postdoctoral research on boy caste fellowship. Professor Prabha is working as professor and head at Department of Biochemistry, the MS University of Baroda. She has been project investigator in number of projects funded from CSIR, DBT, DST, UGC, among many. She is having more than 20 years of experience in academic and research. Professor Prabha research is focused on protein degradation, nanobiotechnology, and biosensor. Professor Prabha served the society by guiding six PhD students. So it is great honor for us to have a legend from the field of macromolecule structure and biology, Professor C. Ratna Prabha. So, ma'am, please, we can start with the today's talk.
Good morning, everybody. My special thanks to Dr. Chigbar, and I thank Rai University for giving presenting this opportunity of speaking on protein engineering. I'm going to start my presentation. Protein engineering is an ever expanding, vast area of research with many different applications. Before we go into protein engineering, I would like to introduce you to protein structure briefly. If we take any protein, it is a polymer of amino acids and it is synthesized in living systems on ribosomes by joining amino acids through peptide bonds. The synthesis starts with the first amino acid for which the amino group remains free, and so we call that as the N-terminus or the N-terminal amino acid, and the amino acids are successively added by formation of peptide bonds between COOH of the previous amino acid and the NH2 group of the incoming amino acid, forming a peptide linkage. And this goes on till the synthesis is completed and mRNA is completely translated. The last amino acid has a free COOH group, and we call that as a C-terminal amino acid or the C-terminus of a protein. The peptide backbone, which is synthesized, folds on itself and takes helix-like and also pleated conformations, and these conformations are cemented or reinforced by formation of hydrogen bonds, and these are called the repetitive secondary structures which are commonly found in proteins. These, in addition to these secondary structures, we also have turn-like structures which are formed when the polypeptide chain changes its direction. So these are called beta turns. Sometimes even gamma turns are seen. The kind of interactions which are seen in a polypeptide chain are, as I mentioned to you before, the secondary structures like alpha helices, the beta sheet, and the turn conformations, the loops. Then there are long distance interactions which involve metal coordination, which involves two cysteine residues forming a cysteine in a disulfide bond linkage, and hydrophobic interactions formed by various hydrophobic amino acids like leucine, isoleucine, valine, etc. And then you also have side chain hydrogen bonds formed between amino acids like serine, which has a hydroxyl group, and aspartic acid COOH group is shown here, but it can be NH2 group of lysine or any other charged group. There's a possibility for hydrogen bonding, and I'm sorry. Then you also have electrostatic interactions between amino groups, charged amino groups and uh, carboxyl groups. These are generally called as salt bridges in the language of structural biology. And these long distance interactions bring distant regions of the protein together, forming tertiary structure. This is the kind of tertiary structure you see in a protein. This is EGFP molecule, and you also see quaternary structure in some proteins where there is more than one polypeptide chain. That means more than one subunit is present, and they can be all polypeptide chains which are present can be of same sequence. In that case, you have a homopolymer or homooligomer, and if you have different polypeptide chains coming together and forming an oligomer, then we call it as heterooligomer. You come across both kinds of quaternary structures in proteins. Before we move further, there is one more concept which is very, very essential, that is domain. Domain is the fundamental unit of tertiary structure. It can be formed by a complete polypeptide chain or a part of it. What it means is, even if you have a single polypeptide chain forming a protein, you can have one single domain or more than one domain. If it is a single domain containing protein, then we call it as monodomain protein. And if you have two domains or more, they can fold independent of each other and they form stable tertiary structures. Then, once again, they may interact with each other or they may remain separate, connected by polypeptide backbone. Proteins can be small with a single domain. As I have told you, monodomain proteins, they would be in the size range of 100 to 250 amino acids, or you may have a protein with many domains, then the size will be much larger. In such a case, the polypeptide can be cleaved into individual domains. And the domains, when they are cleaved, they can fold on their own. And not only that, they may be associated with a function that can be accomplished independent of rest of the protein. So here I am showing you a protein. This is ornithine decarboxylase protein. And if you can cut the protein, you can see two domains here. And if you can cut the protein in between, you would be able to release a domain like this. Okay, even this domain can be released separately. To illustrate, I am just showing one domain. Now, if we look at proteins, protein evolution, how so many proteins have come into existence. Nearly 30,000 proteins are known to be present, and uh, they have come into being. And one more important point is, if you take dry weight of a cell, 40 to 50% of the cell's dry weight is made up of proteins. Proteins are also known to carry out many functions. Nearly 10 different groups of functions are seen in any standard textbook on proteins. And if we look at the functions, two very important functions are, they act as enzymes and they act as machines, basically allowing you to move and do a lot of actions. So they are often called as workhorses of a cell or molecular machines by different people. Divergence of duplicated sequences by mutations, deletions, and insertions, which are formed during the process of speciation. Whenever a new species comes into being, it would have, say, if we take even a common protein like hemoglobin, whenever two species are diverging, the sequence of hemoglobin changes, or cytochrome C. It is very important. It is present in the lowermost organisms to the highly evolved. All of them have cytochrome C in them, but the sequence is only partially conserved. The rest of the residues have changed. And the process of this change happened because of mutations, deletions, and insertions of new residues. But the function is retained. So if it is a cytochrome C in, say, amoeba, it is cytochrome C even in a human or in a horse. And then you have another group of proteins called paralogs. These also evolve along the same lines, but previous to that, there is a gene duplication event. One of the copies of the genes remains with the original function, and this other dispensable copy starts accumulating mutations, and this gives rise to new proteins with newer functions. Okay, so here we can take the example of, say, lysozyme, which acts against bacterial cell wall, and alpha lactalbumin, which is a milk protein. Both of them have 40% sequence homology, and almost same kind of structure, but entirely different functions. So this group of proteins are called paralogs. Then comes the most important process of evolution of proteins. That is, recombination of genes results in new arrangement of domains and novel proteins with very different functions. This is how 70% of prokaryotic proteins have been formed, and 90% of eukaryotic proteins account for this domain shuffling as a result of gene rearrangements. This is an example how the process of domain shuffling occurs. Here you have some geometric shapes, each of them representing one particular domain in a protein, and you can very clearly see that carbamoyl phosphate synthase large chain has a domain which is similar to that of biotin carboxylase, glutathione synthase, and it has another domain which is common even with succinyl-CoA synthetase beta chain. Like that, several domains have come together and this particular protein has formed. In this process, you would find that substrate binding site of one protein is going to join with the catalytic site of another protein, giving rise to an enzyme with the catalytic function of the second protein and substrate binding function of the first protein. This way, you have a completely new enzyme evolving. So, uh, you may wonder, what has protein evolution got to do with protein engineering? When Dr. Chibber asked me to present a talk on protein engineering, incidentally, I was reading a novel, Origin by Dan Brown, and there I came across a quotation by Charles Godey, which says, "Nothing is invented, for it's written in nature first. Originality consists in returning to the origin." And I found that this is very much applicable to protein engineering. Whatever is happening in protein evolution is kind of adopted in protein engineering. Protein engineering has a striking similarity to protein evolution. Both obey the basic principles of protein structure. You would not have Instead of alpha helix, something completely different. Maybe instead of a right-handed alpha helix, you cannot make a left-handed alpha helix and have a protein functioning in the cell normally. Okay, so you have to stick to the same rules, and that is so even with protein evolution. Okay, and also they follow the same phenomena. What I mean to say is you would have insertions, deletions, substitutions of amino acids or stretches of amino acids. In addition, you would have truncations, you would have insertion of new domains and recombination of domains, which is called domain reshuffling. These kind of things would give rise to completely newer proteins. However, there are two main differences between them. Protein engineering is much faster. It is carried out with a definite purpose. It because if you look at protein evolution, it will take several eons before a functional protein emerges from an ancestral protein. Whereas when you are doing protein engineering, you want to meet the goal within your PhD time. So it has to be much faster, and it is also carried out with a definite purpose. You have already some hypothesis, and you are working on that to make a protein of that particular nature meeting a particular purpose. The many purposes of protein engineering as we see it today are creation of novel proteins, manipulating the stability of proteins with respect to temperature, pH, and other conditions. This is important, especially when we are working with enzymes and we have some biotechnological purpose in mind. When we are working with fermenters, etc., your protein should be stable at a given temperature, pH, and other conditions. You cannot have your enzyme separately and maintained at the optimal temperature and pH. Then creation of novel antibodies and other proteins for therapeutic purposes. Novel of therapeutic proteins is your insulin expression, antibodies are some of the examples. Novel antibodies for biosensor applications and affinity applications. Then you can also have fluorescent proteins for cell biology studies and transporting across membranes can be made possible. Some of you might have heard of TAT binding protein, TAT proteins. They these are HIV proteins, and they have a sequence with many positioned successively, and these proteins or this particular sequence transplanted on any other protein, the protein is transported across the membranes. This is a very interesting application whenever we want to send some protein across the membrane to solve some intracellular problem. Then understanding structure function relationships in proteins. Then we also can look for targeted protein degradation. There are many intracellular and intercellular aggregates of proteins formed, especially in aged population. If we can come up with a solution, then we would be able to clear these protein aggregates and probably treat the diseases successfully. Protein engineering can be approached from two different routes. One is chemical method, the other one is gene manipulation. Chemical methods used for protein engineering are some of the things you must have seen commonly. Treatment of hair with mercaptans for curling or straightening in hair styling, Then treatment with DEPC, diethyl pyrocarbonate, revealed that prions are tenacious in nature. The substrate of trypsin can be restricted because you know that trypsin cleaves in two positions. మళ్ళీ స్టార్ట్ చేద్దాము. హలో హలో presentation. How do you see the okay. Pa- Pardon? From the beginning. Can you repeat? Hello? ఎందుకు అయిపోయింది ఎక్సెప్ట్ అండ్ ఏమైంది నాయిస్ వస్తుందా Hello. Unmute just. Hello. Can you hear me? Hello. Can you hear me? Yes, sir. Now everything's fine. Perfect. Okay. I start. So limitations somehow led us to the limitations in webinars, and we have overcome successfully. Sorry for the interruption. Now coming to the presentation, limitations of chemical methods. However, protein engineering by chemical methods, like treatment with chemicals, is non-specific. So, for example, if we are treating lysines, we cannot treat one particular lysine. We end up modifying all the lysines present in a protein. Sometimes our need may be limited to only one lysine. So the best way to attack such a problem is to do modifications at genome level. And mostly it is mostly suitable to in vitro studies. That is, only when you take the protein out, you would be able to treat it with a chemical. Very, very rarely it will be possible to treat with a drug when the protein is inside the cell. And it has to be carried out in a batch to batch manner. So if we have isolated the protein, modified it, it has served its function. Once again, you are back to do the same reaction with the next batch of enzyme purified. So these limitations would not let us adopt chemical methods completely for protein engineering. So the other alternative is to go for protein engineering by gene manipulation. By substitution of one or more amino acids, we would be able to do that. By deletion or insertion of one or more amino acids, by truncating a domain, by adding a domain to another protein, by interchanging domains of proteins. These are the different alternatives we have at hand. Alpha complementation is one thing everybody who had some molecular biology experience must have come across sometime. Beta galactosidase enzyme, which is encoded by LAGZ gene, is a tetrameric protein. It is a homotetramer, and the protein is expressed from the genome of E. coli and other organisms also. So LAGZ of E. coli somehow underwent mutation, and it started expressing a protein lacking N-terminus. And it was found that instead of a tetramer, it was forming dimers. And then when they saw this in the year maybe 1967, what they had done was that they had added N-terminal peptides, which were obtained by tryptic digestion of the whole protein from an external source. And this is a secretory protein. So the two assembled together and they got a functional enzyme, and this functional enzyme was found to be a tetramer again. You can see this, it's a tetramer. Initially it was a dimer, where when genomic DNA underwent a deletion, what they got was a dimer. And when they had added these tryptic peptides of N-terminal, they got the functional enzyme back. And this functional enzyme could act on your XGAL and release blue color. And then what they had done was they had taken this strain and introduced a plasmid which could encode the N-terminal region of the protein. So now they have the C-terminal part of the protein as well as the N-terminal produced within the same cell, and it was assembled and the functional protein was produced, and this functional protein worked on XGAL to give you blue colonies. Then later on what they did is, all of you know that they have inserted a multiple cloning site in this N-terminus encoding region, and if you clone any gene there, the N-terminal peptide that would be produced from this region is disrupted because of the presence of the gene, and it gives rise to white colonies. So this blue-white screening has become an easy tool for you to detect whether your clone contains the gene of interest or not. Okay, this is a primary screening technique which has been adopted, and we refer to it as alpha complementation of beta galactosidase. I also mentioned that for biotechnological purposes, the proteins which are available in nature may not be able to function in the pH conditions and temperature conditions we can offer. So we had done some work on Proteus vulgaris lipase. Nature has evolved and designed enzymes to perform different tasks. But in the pursuit of biotechnological interests, the enzymes must often be improved, altered, or even completely redesigned. So this is a Proteus vulgaris lipase with which we were working, and rest of the experiments have to be performed at a higher temperature, and this was affecting the enzyme. The enzyme was performing poorly. So we thought that if we can increase the strength of the structure, we reinforce the structure by introducing a disulfide bond, then the protein would be more stable and would not be affected by temperature. So what we had done was, we had taken a glycine 131 and threonine 238, changed these two residues to cysteines. As I had shown you earlier, if two cysteines are present in proximity and the conditions are favorable for disulfide formation, that is the redox potentials, then they would be able to form a disulfide bridge. And this secretory protein formed a disulfide bridge. Another interesting point I would like to mention here is, either the membrane proteins or the secretory proteins contain disulfides. Generally, cytosolic proteins would not be able to have disulfides instead of having many cysteines, because the redox potentials inside the cell, the reducing environment which is present inside the cytosol, would not favor disulfide formation. So what we had observed, since it is a secretory protein, we had no problem. It formed cysteine, and the T half of the protein increased from 37 degrees to 50 degrees Celsius. The reaction with non-edible oil could be completed in 30 minutes instead of 24 hours, what it was taking earlier. So this was one way in which protein engineering could be used successfully to get a better enzyme. Proteins have an interesting feature in their structure, especially those proteins which act as cell switches. There are some kinases in the cell which would take the cell towards a particular direction. They generally belong to the signal transduction pathways. These kind of enzymes and cyclins, which are necessary for cell cycling, they have to be removed as soon as they serve their function. Any regulatory protein or cell switch has to be removed once it has served its function. And it should be replaced with a set of enzymes or set of proteins which are required for the next stage. If that does not happen successfully, then you would have a disease condition like cancer, apoptosis, etc. So it has to be very strictly regulated, and features or sequences of proteins that confer metabolic instability are called degradation signals or degrons. So such proteins contain certain features which would make the sequence which would make the protein unstable. The first degradation the first degron to be discovered is a destabilizing N-terminal residue of a protein, or the N-degron. A set of N-degrons containing different destabilizing residues yields a rule which is termed as NN rule. NN rule relates to the in vivo half-life of a protein to the identity of its N-terminal residue. NN rule pathway is present in all organisms from bacteria to mammals. Now we will see NN rule pathway. Here what they had done was a protein engineering experiment using site-directed mutagenesis. Here ubiquitin fusions. Ubiquitin is a small protein which is present ubiquitously. So they have taken the gene of ubiquitin and fused it with beta-galactosidase gene, and the N-terminus codon of beta-galactosidase was changed such that 20 different variants were made, and each of these variants has a different codon like arginine, lysine, histidine, phenylalanine, leucine, tryptophan. This list you can see here. All 20 amino acids are present here, and here you have the ubiquitin encoding region, and here you have beta-gal encoding region. This whole thing is expressed as a chimeric protein in the cell. When this is expressed as a chimeric protein, UB-specific C-terminal hydrolases come and cut right after UB, releasing, that is UB means ubiquitin, and release the beta-galactosidase molecule with different N-terminal residues. And then they monitored what is the half-life of these variants of beta-galactosidase. And they found that these proteins had different half-lives. And interestingly, previous to doing this experiment, they had looked at the different proteins in a cell, and they found that different proteins were having different half-lives. This actually led to this particular experiment, and they found that what they had observed earlier with different proteins matched with the beta-galactosidase half-life. When, say, arginine is present, then they looked at the half-life of another protein which has arginine as the first residue, and it led to formulation of the N-degron rule. So here you can see the relation between N-terminal residue and half-life of the protein. And if you have arginine, lysine, and histidine, all these three positively charged amino acids, or you have bulkier hydrophobic amino acids like phenylalanine, leucine, isoleucine, then what is observed is they have very short half-lives. Whereas if you have aspartate and glutamate, the acidic residues, then also you find that the proteins have shorter half-lives. Once again, aspargine and glutamine also showed shorter half-life. Whereas the other residues like cysteine, alanine, serine, threonine, glycine, valine, methionine, all of them led to longer half-life. In fact, the protein synthesized in the ancestral generation would be seen in some 2-3 generations later in the daughter and granddaughter cells. Here you can see the identity of different residues and also which are primary destabilizing residues. As I told you earlier, all the bulkier hydrophobic amino acids and also the positively charged amino acids, they would confer shorter half-lives and they are called primary destabilizing residues. Then you have aspartates and glutamates, the acidic residues, they also confer shorter half-lives. And these, when a protein has these residues as the first or the N-terminal residue, an arginine is transferred to the N-terminus, making it destabilizing. So these are called secondary destabilizing residues, followed by glutamine and arginine, glutamine and aspargine. Glutamine and aspargine are the amide containing amino acids. The amide groups are hydrolyzed by an enzyme and glutamic acid and aspartic acid are formed. So from tertiary destabilizing, these amino acids give rise to secondary destabilizing amino acids by the action of an enzyme. And then finally, arginine is fixed before, so the protein is taken towards degradation. So you have 3 groups of destabilizing amino acids: primary destabilizing amino acids, secondary destabilizing amino acids, and tertiary destabilizing amino acids. Any other amino acid which doesn't belong to any of these 3 groups would be classified by default into stabilizing amino acids. As you know, most of the proteins inside the cell are synthesized with methionine as the first residue. Now, this slide once again shows you what are the primary destabilizing residues, and there is you have tertiary destabilizing residues, anti-amidase, that is N-terminal amidase, comes and acts on these tertiary destabilizing residues, and aspargine and glutamine are converted to aspartate and glutamate. Then R transferase comes, acts on it, fixing arginine before these 2 residues and forming the primary destabilizing signal. Rest of the amino acids like RKH, that is arginine, lysine, histidine, leucine, phenylalanine, tyrosine, isoleucine, tryptophan. So all these are primary destabilizing residues, and they would be taken, and these proteins are ubiquitinated and degraded by ubiquitin dependent pathway of degradation, and proteasomes act on them to break them into pieces. So another group of proteins, the moment you see these Y-shaped structures, you know that they are antibodies. And in this COVID times, antibodies are getting discussed almost every day, and they have become a household topic. Now, what you are seeing in the center is an IgG. This IgG molecule has 2 light chains and 2 heavy chains. The 2 light chains have 2 variable and 2 conserved domains, or 1 variable, 1 conserved domain per chain. And if you look at the heavy chains, per chain, you have a variable domain at the top, followed by 3 conserved domains. And the 2 heavy chains are connected by disulfide bridges. Again, the light chain and heavy chain are also connected by disulfide bridge. When this antibody is subjected to digestion by papain, it would release two different kinds of fragments, the fragment and the FC fragment. And if the same thing is digested by pepsin, it would release FAB prime and FC prime fragments. These fragments are put to various uses many times on antibody columns. There are many different applications which are seen in the affinity purification, and in therapeutic area also they are used. And then they came up with the novel idea called diabody. Diabody is having two different epitope binding sites. The first epitope binding site binds to one antigen, and second epitope binding site binds to second antigen. These are now made by several companies. These are camel group of animals. They are called camelids. You can see dromedary camel, which is seen commonly in India, especially in Gujarat and Rajasthan. And you have Bactrian camel, which is two-humped. Then you have Vikurna, and you have alpaca and llama. Most of these animals are residents of the American continents, and especially the South American continent. The two-humped camel, as you can see, a pyramid in the background, is found in Africa. Okay, so why we have diverged all of a sudden from the discussion on protein engineering is these group of animals have one very special kind of antibody. You can see the IgG antibody. Just now we have discussed IgG antibodies, and here you can see this is a single chain antibody which is formed by cutting this region out and introducing a peptide chain connecting them. Okay, this is a genetic manipulation. Actually, at genomic level, you have to take this gene and then link up these two fragments of DNA with another fragment of DNA so that they remain connected, and this gives rise to a single chain antibody. Okay, now in continuation with the discussion on camelid antibodies, here you can see that these are having only heavy chains. There is no light chain here. And in one case, this junction which is present between the variable domain and the conserved domain is too long. Okay, and here you can see that one of the domains is missing, and same here, but it is only that the connecting peptide is shorter. Okay, these two antibodies can be cleaved here to release this region. These fragments are called nanobodies. Nanobodies find many more applications, even more than single chain antibodies, as these are smaller, they are narrower, so they would be able to go and penetrate and bind to the epitopes which are present in crevices, and they're less cumbersome to handle. You can have them on biosensors. You can also use for therapeutic purposes. You can use them for affinity purifications. So there are many different applications seen for these nanobodies. Coming to humanized antibody, this is another interesting thing that is happening in the field of immunology. What they have done is, if you have raised antibody against an antigen in rat, mouse, or rabbit, you would be able to produce antibodies. And when you are injecting them to humans, Many times there would be immune reaction in human system. So to overcome that, if you can have a hybrid antibody, only the epitope binding region is taken from, say, rat or mouse, wherever it is produced, and rest of the antibody structure is similar to that of human. That is, you take a human gene and just exchange that portion which corresponds to epitope with that of rat or mouse from which the antibody is got. So in that case, the humanized antibodies are got. These are effective in fighting the pathogen. At the same time, they would not give rise to immune reaction in the individual to whom they are administered. Here, we did some work on targeted protein degradation. Some proteins are specifically targeted for degradation. They have degradation determining sequences inherently present in them. The degradation determinant signals are called degrons. So you may wonder that we had already talked about N-degron. That was only the N-terminal residue. Here we are addressing a whole sequence which is present inherent to the sequence of the protein. Somewhere inside the protein, you would come across this sequence. The degron sequences can be transplanted on other proteins. The receptor proteins with degrons can be targeted for degradation, and this degradation can be regulated with the help of certain molecules. And for this particular work, we chose ornithine decarboxylase. Ornithine decarboxylase is an enzyme which is required for polyamine biosynthesis. And this is the first enzyme of the pathway and also the key enzyme. So you have feedback regulation happening at the level of ornithine decarboxylase. And ornithine decarboxylase is regulated by more than one mechanism. It comes and binds to ornithine and converts it to putrescine by releasing a carbon dioxide molecule. You can see the reaction here. This is the ODC enzyme, and ODC enzyme is a dimer, homodimer, and this particular ODC dimer is in dynamic equilibrium with ODC monomer. At any part of time, 40% of the molecules are in monomeric state. When the polyamine levels rise, polyamines are molecules which go and bind to nucleic acids and regulate the process of transcription, of replication, etc. So, polyamine levels have great significance with respect to taking the cell either towards cancer or towards apoptosis. So their levels have to be strictly regulated. Hence, the regulation is operated with great care inside the cell. And as I said, there is a dynamic equilibrium. And when the polyamine levels rise, another molecule, another protein called antizyme is induced. This antizyme goes and binds to ODC monomer, forming a complex of ODC antizyme. This binding of ODC antizyme starts driving the equilibrium towards ODC monomer formation from dimer. And this monomer is degraded as antizyme delivers this to 26S proteasome. 26S proteasome degrades ODC, and since ODC will not be available for fresh polyamine biosynthesis, polyamine levels are brought down. Okay. Now, this is what is proteasome. Proteasome is a protease present intracellularly. This is present in cytoplasm, in the nucleus. And this proteasome is made up of two different kinds of subunits. Alpha subunits forming two outer rings 7 subunits make one ring, and 7 beta subunits form 2 inner rings. Each subunit contains each ring contains 7 subunits. So you have a 4-tiered system where 2 outer rings of alpha nature and 2 inner rings of beta subunits, and this is a hollow cylinder. So any protein that enters from here would be degraded and the peptides are released. Here, the beta subunits, 3 of the beta subunits out of 7 have trypsin-like, chymotrypsin-like, and caspase-like activities present in them. Okay, and the outer 2 rings serve as antechambers. They would permit the entry of only those proteins which are destined for degradation. How would this system know which protein needs to be degraded? So the system will come to know a protein needs to be degraded by recognizing a signal present on the protein. In this case, it is antizyme, and there is an assembly of regulatory subunits present on both sides of this cylindrical protease like this. These subunits of regulatory body that is called 19S will bind and recognize the signal, then remove the signal from the protein which needs to be degraded, and the protein is unfolded and threaded into the tunnel of the proteasome, where it is degraded with the help of protease subunits which are present on the inner cavity of the proteasome. This is how the peptides are released. So this is the structure of the enzyme ornithine decarboxylase. It has 2 subunits, the alpha beta domain and a C-terminal beta sheet domain. In case of yeast, it has an N-terminal overhang of 50 residues here. And in mammalian system, the N-terminal overhang is absent, but it has a C-terminal 35 to 37 residues tail present. We made a comparison of the proteins of yeast ODC and human ODC. And as I told you, here we see the overhangs, the N-terminal overhang of yeast ODC, and followed by the alpha beta subunit. And this is the beta sheet domain. Here in human ODC, you have N alpha beta present here, followed by beta domain, beta sheet domain, and then you have a C-terminal tail. What we had hypothesized is if we would be able to remove peptides and attach those peptides to target protein, some reporter protein, then we should be able to see degradation if the peptide contains a degron. Okay, so we have decided that we would cut out 1 to 50 residues because it is acting as a separate domain. Then 50 to 346, another domain which is alpha beta domain, and the first 50 residues joined to alpha beta domain, which we called as N alpha beta, and then the C terminal belonging to mammalian protein, that is 425 to 461. These are the regions we selected and added to a reporter protein. In this case, initially we started with GFP. Then we looked at the GFP levels when Mg132 is added to the system. That is, Mg132 is a proteasome inhibitor. the question we asked is whether these proteins, chimeric proteins, having the degrons, are degraded by proteasomes or by some other mechanism. If they are degraded by proteasomes and proteasomes are inhibited, then there won't be any degradation. So we found our answer this way: control where there is no where there is ODC, where there is GFP, and we found the GFP being present in both. And here what we have observed is when there is no MG132, the GFP got degraded, and MG132 is added, proteasome is inhibited, then the protein remained intact in spite of the degron. That means degron is required for its degradation by proteasome. Then alpha beta domain, whether MG132 is present or not, is showing the same levels. That means it is not degraded by proteasome. And in case of N alpha beta, once again MG132 is not there, then we could see better degradation compared to when MG132 was there. Then we wanted to see what would be the effect of antizyme, whether antizyme can bind all these peptides and take them for degradation or not by varying the concentrations of polyamines. As I told you before, when we are adding polyamines to the medium, then excess polyamine would be inducing the expression of antizyme, and we have found that there is no change in N50. That means N50 is not recognized by antizyme. Whereas with N alpha beta, we found a steady decline in the level of the protein, reporter protein, and that means this degradation is assisted by antizyme. So somewhere in the N alpha beta, antizyme is binding. Then there is no change at all in the alpha beta level. That means it is neither degraded by proteasomes nor antizyme is assisting its degradation. Then what we had done, once again, this is monitoring fluorescence by another technique, and we didn't see any change with alpha beta, with N50, whereas in case of N alpha beta, when antizyme was present, there was greater amount of degradation. And this is a Western blot analysis to support the same thing. And here what we had seen is, again, GFP levels in the cells at different time points and with MG132. What we have seen is where there is inhibition, the level of GFP increased in N alpha beta and N50 because of the inhibition, whereas in alpha beta there is no inhibition. So the degradation occurred by other mechanisms. Again, same experiment we had done with beta galactosidase, as they wanted to see if it is because of GFP the degradation was occurring. This was our peer reviewers' comment. So here we had used beta galactosidase and showed that N alpha beta caused degradation by proteasomes. Once again, N50 also caused degradation. Then we recorded the structure of these peptides using CD spectra. And this was important because if antizyme or proteasome have to recognize them, they should be able to maintain the structure. What we have seen is that N alpha beta was adopting a structure which is native like. Okay, and 40% TFE was added to see if it can induce more helicity in the molecule. And this is alpha beta domain, means it has alpha helical structure as well as beta sheet structure. Since there is lot of alpha helix, we had added 40% TFE to see if the helix formation is complete or not. And it's already complete, there's no change, and the structure is almost native like. Then coming to alpha beta, we could see only beta sheet like structure. This hump like structure, which, or the trough like structure, which is seen between 208 to 218 nanometers, is an indicator of beta sheet structure, but it doesn't show any helicity. Then coming to N50, N50 initially did not show much helicity, but addition of 40% TFE gave rise to helicity in the molecule. Then we looked at the CD spectra and analyzed using CD Pro software. What we have found is the amount of helicity in N alpha beta is higher compared to alpha beta, and also N50, which is reported by many previously that it doesn't have any structure, appears to adopt a 310 helical conformation. Then we looked at the sheet content. Alpha beta has greater sheet content compared to N alpha beta, and turn structure and other loop like structures were also obtained. These are the other percentages. Now we can compare these results with what we got with using ITASR, and this gives you what is the percentage of structure in the native protein. Native protein has 37.6% helicity, and N alpha beta shows 30.8. So they are closer, whereas alpha beta shows only 17.7, that is half of it, less than half of it. And N50 region has 52% helix. Here, by adding TFE, we could get something like 32.3% helicity. Comparing to the sheet values obtained in CD with those that were obtained theoretically, looking at the sequence, is 10% in the case of N50, which is closer to 16.6 what we had seen, and helix sheet content in N alpha beta is 16.1 and 18.6 what we got here, and alpha beta shows 18.1, whereas here it is 27.9. Okay, so we can say that alpha beta could not take up, by itself could not take up native like structure, because of which it is not recognized by antizyme and it cannot act as a degron at all. The N50 region is unfolded region, which is necessary for the proteasome to let the protein into the tunnel and carry out degradation, which is seen with N50, and it acts as a degron by causing degradation. But in case of N50, since there is no antizyme binding region, the degradation is not regulated. Coming to N alpha beta, in N alpha beta, the helix, sheet, both are very close to that of native state, and it can be recognized by antizyme which then delivers this target protein along with N alpha beta for degradation to proteasome. So the conclusions we have drawn after doing this work were N50 and N alpha beta can act as potential degrons. N50 and N alpha beta adopt native-like conformations. Alpha beta cannot act as a degron as it cannot fold to native-like structure. Polyamine surplus induces antizyme. Degradation of N-alpha beta was regulated by antizyme expression. This regulatory feature makes it an ideal degron for targeted protein degradation. What we mean by this is if we increase polyamine concentration in the medium, antizyme levels will increase and there would be increased degradation in case of N alpha beta. And if you bring down the polyamine levels, the the antizyme levels will also decrease as a consequence and overall degradation comes down. So this way it can be used to regulate the degradation. Then degradation activity of Degron N50 is not affected by antizyme expression as it lacks antizyme binding element. So polyamine concentrations would not affect degradation in any way with N50. So this work was published by us in BBA General Subjects. This is the model we have proposed after doing the work. This is the chimeric protein with alpha beta domain and EGFP. Degradation by proteasome does not happen. This is the N alpha beta domain and we have GFP here. There's degradation happening, and if you increase polyamine concentration, there would be better degradation. The last one is N50, and N50 leads the protein for degradation by proteasome. So N50 and N alpha beta can act as degrons, and N alpha beta acts as a regulatable degron. The other interest of ours, which also involves protein engineering, is studying structure-function relations in ubiquitin. Ubiquitin is a highly conserved protein. It is present in all eukaryotes. And if you compare the sequences of ubiquitin from yeast to plant or yeast to human, you find that only 3 residues have changed from yeast to any of these organisms. So there is almost 96% conservation of sequence of ubiquitin. The gene family, ubiquitin is an essential gene. Hence, the gene is presented in many copies inside the cell. There are at least 4 copies in yeast. The first 3 have single copy of ubiquitin. The last one, UBI4, is a gene fusion having several copies of ubiquitins present in succession. And all the ubiquitins are expressed as fusion genes. First 2, that is UBI1 and I2, are expressed as large ribosomal fusions and they have 40A subunit fused to them. UBI3 has S31, that is small ribosomal subunit fused to it. As I told you, UBI4 is a gene fusion of ubiquitins. All these ubiquitins are transcriptional fusions, and once translation happens, the fused product that comes out is quickly separated into 2 proteins or independent proteins with the help of UBC terminal hydrolases. Ubiquitin by itself can serve many functions when it is attached as a post-translational tag to other proteins. But if you look at catalytic activity of ubiquitin, it has none. It is not a catalyst, it is not an enzyme. It is used as a tag for post-translational modification of other proteins. So if you have monoubiquitination occurring, many times it helps in trafficking and endocytosis. Monoubiquitination can happen on a lysine side chain. The way ubiquitin is added is C-terminal COOH of ubiquitin is added to the epsilon amino group which is present in lysines, and an isopeptide linkage is formed. Sometimes ubiquitin is also joined to the N-terminal of methionines. That is first methionine. Then you have 7 lysines present in ubiquitin, and all of them are active. They encode one function or the other, and these ubiquitins are added on K6, K27, K29, K48, and K63, and also K33. These are the places where you would find ubiquitins, and all of them can undergo ubiquitination. That means one ubiquitin is added to the protein, and the lysine present in the ubiquitin is once again added with another ubiquitin molecule. Lysine in the incoming ubiquitin once again is modified by a third ubiquitin molecule. This way a polyubiquitin chain is built. For this polyubiquitin chain formation, K6, that is lysine 6, lysine 29, lysine 48, and lysine 63 are more commonly used, but all other lysines are also used to the same extent. So they have all these functions, regulatory functions, and proteolysis by proteasomes, and also DNA damage tolerance, kinase activation, trafficking of molecules across membranes, and regulating translation. You know that histones also undergo ubiquitination. That is how they regulate transcription. And once ubiquitins are added to a protein, and the protein is taken for degradation or it has served its function, the ubiquitins which are present in the polyubiquitin chain are disassembled and recycled again to form fresh ubiquitin chains on newer molecules. The process of ubiquitination is catalyzed by a cascade of 3 enzymes. The first enzyme comes and the first enzyme, which is called E1, binds to ubiquitin which is present in the cytosol and activates it in an ATP dependent reaction. This activated ubiquitin is transferred to the second enzyme E2, and E2 goes and binds, charged with ubiquitin, it goes and binds to another enzyme, ubiquitin conjugating enzyme E3. Hello? Almost towards the end. You have some time limit? Okay. Yeah. Okay. Then this E2 charged with ubiquitin comes and binds to E3. Then from E2, ubiquitin is transferred to substrate. A polyubiquitin chain is built this way. And polyubiquitinated protein many times is taken for degradation to 26S proteasome. Ubiquitin molecules are freed and peptides are released. Here, what the question we asked was that when ubiquitin sequence is so strictly conserved, then all the residues which are present in ubiquitin must be playing some role either in structure or in function or in both. So to answer this question, what we had done was we subjected ubiquitin gene to error-prone PCR, and the mutant forms of gene that were generated in the reaction were cloned into the plasmid. And then we introduced the plasmid into the cells and screened for mutants. What we found is it was one of them turned out to be a dosage dependent lethal mutation. As you increase the concentration of inducer, the cells were dying. So this we found was very interesting. We had gone for sequencing and we found that there are 4 places. S20 was converted to F and A46 was converted to S and L50 was converted to P. I61 was converted to T. Okay, where do they occur in the sequence is S20F. This is the third residue of a type 1 turn. These substitutions did not change the residue in these cases. So you would not see any effect at protein level. Now coming to, these are means synonymous codons. So coming to the last one, coming to the next one, alanine 46 has been changed to serine. This is the second residue of type 3 turn. Leucine 50 has been changed to proline and this is part of beta sheet. Then the last one is isoleucine 61 has been changed to threonine, which is present between 2 turns in the turn-rich region. As I told you, this is a UBEP42 is a dosage-dependent lethal mutation. This conclusion we had arrived at by increasing the inducer concentrations. The gene for ubiquitin after mutation was cloned in a plasmid which has a CUP1 promoter, which is responsive to copper sulfate concentrations. If you increase copper sulfate levels in the system, there would be more and more transcription happening from this promoter. And we have found that when there is 0 micromolar copper sulfate, all cells were growing normally. Then we increased copper sulfate concentration. We found that UBEP42 level started reducing and finally they disappeared. So when we found that this is a dosage dependent lethal mutation, we wanted to see if all 4 of these mutations are behaving alike and contributing to this lethal phenotype, or this is a cumulative effect, or some of them are responsible for the lethality seen. So we decided to separate them and express them, and the result we got is among these S20F and A46S are not lethal, but L50P and I61T turned out to be lethal. Once again, dosage dependent lethality is seen. Then we had grown the cultures, submitted them to FACS analysis to see which part of the life cycle is getting affected. And in the cell cycle, there is an arrest happening in the G0, G1 phase. In case of L50P, I61T, and UBEP42. All others are growing normally. Then we looked at cyclin-dependent kinase levels, which is in CDC28. Its levels are important for cell cycling. And we have found that UBEP42, UBL50P, and UBIC61T were giving reduced levels of CDC28. There is a MAP kinase, FUS3. We looked at FUS3 levels. FUS3 behaves exactly opposite to CDC28, and we have seen that UBEP42, UBL50P, and UBI61T were showing elevated levels of the protein. Then we looked at mRNA and we found that there is no change in mRNA levels. Whatever change is happening is at protein level. So we once again asked a question, if we give MG132, which is an inhibitor of proteasome, what would be the effect? This will tell us whether the protein is degraded by proteasome. Then what we have seen is that even when MG132 levels are less or absent, these proteins, L50P, I61T were higher, and the proteins S20F, A46S were lower, and for FUS3, and here CDC28, UBEP42, L50P, I61T showed lower levels, and the effect changed with MG132 completely. So this indicated that UBL50P, I61T, and EP42 are degraded by some other protease, not by proteasomes. They are inhibitory to proteasomes. Then we looked at cell sorting, heat stress effect. And what we had seen is that once again, L50P, I61T, and EP42 are detrimental for the cells. Then we looked at uracil permease, which is present on the membrane, and it is degraded by endosomes. And here what we have seen is this particular process gets affected in L50P, I61T, as well as UBEP42. Then we looked at cell sorting of CPS. Once again, for the UBEP42, L50P, and I61T showed the effect on cell sorting. We looked at antibiotic resistance. Here, once again, ubiquitin mutations affected antibiotic resistance with respect to hygromycin B and tunicamycin. We looked at polyubiquitin chain formation. It is happening all right, but if we look at K63 dependent polyubiquitin formation, EP42, L50P, and I61T mutations reduced K63 dependent ubiquitination. And this K63 dependent ubiquitination controls cell sorting, which is seen affected. It also affects the antibiotic resistance, and we have seen it is affected. Polyubiquitin formation and K63 ubiquitination, once again we have seen the same effect. Now coming to CD spectra, we found that UBE42, EP42, UBL50P, and I61T cannot maintain native-like structure. These mutations have drastically affected structure formation in the protein. So these are the conclusions what I have already explained. The mutations EP42, L50P, I61T are dosage dependent lethals. Uh, then transformants expressing L50P and I61T displayed slow growth like UBEP42, and L50P I61T affected cell cycling as CDC28 levels reduced. EP42 L50P I61T showed elevated levels of MAP kinase FUS3. Then CDC28 and FUS3 are not degraded by proteasomes. In spite of that, there is an indirect effect produced by ubiquitin mediated mutations, impairment in degradation of proteins by UFD pathway, which I have not talked to, then L50P I61T affected survival under heat stress. Uracil permease degradation by lysosome was also affected in these three mutations. Peptidase S could not be sorted out with these mutations being present into the lysosomes. Then survival under antibiotic stress was also affected. The mutant forms could not be incorporated into K63 polyubiquitin chains. So these are the observations we had made. So this is how we can make use of protein engineering to study structure function relationships. As I have told you, we can make an enzyme better and more suitable for the biotechnological purpose we have in mind. We can have therapeutic antibodies. We can have antibodies better suited for applications like chromatography, like immune reactions. We can also have targeted protein degradation. So there are so many applications, and the number of applications keep on increasing as the field is expanding, and the field has great potential for any young researcher to take up. So with these things, I thank the organizers for giving this opportunity. Okay, thank you. If you have any questions.
So thank you, ma'am, for such an informative session.
Yeah, thanks.
So we are having a few questions from the participants. So first
Okay.
question is, why does the mechanism of pepsin and papin differ, and how come one gave two fragments and the other gave three fragments respectively?
Okay. Pepsin recognizes residues which are bulky hydrophobic residues and cleaves there. Okay. And papain is a cysteine protease and it recognizes different residues. So that is why, depending on the sequence, you would get different cleavage patterns
Okay,
depending on the sequence of the antibody.
okay. No, thank you, ma'am. Another question is, can we stop the spread of coronavirus by blocking the spikes of SARS-CoV-2 virus? with the help of engineered camelid antibodies, as they are small and can access the antigen easily.
This is one possibility which can be looked into. Probably somebody is doing it. I'm not fully aware, but definitely
Okay.
there's a possibility.
Okay, okay, nice. Thank you, ma'am. Another and the last question is: does the change in residue of ubiquitin affects its function in different organisms?
Pardon?
Does the change in the residue of ubiquitin affects its function in different organisms?
See, I have not presented some part of my work in which we have done S63D mutation.
Okay, okay,
Okay. And in yeast, we had not seen any serious effect with S63D mutation. Later on, somebody else pursued the same work with S63E mutation. Okay, D is
okay,
S is serine. Serine replaced by aspartic acid we had done and reported this work in maybe 2012. We published it. 2011, 12, I don't remember the date. Then
okay,
somebody else published similar work, that is S63 serine mutated to glutamic acid. But this work was done in human cells, and they said that this particular mutation has some neurological consequences, whereas in yeast system we did not find. And D and E are somewhat homologous in structure, aspartic acid and glutamic acid both are acidic amino acids. Okay, so you
okay.
can anticipate similar
Yes.
effect.
Okay, okay. So thank you,
Yeah,
ma'am, so much for answering all the queries by the participants, and I
yeah.
hope that this session is highly informative to all the researchers or in the academician who are working in the field of protein engineering. And now I would like to call our Dr. Assis Rami, Assistant Dean of Center Research and Development and the head of Rai Business School, to kindly deliver the vote of thanks.
Yeah, thank you very much, sir. It is my privilege to propose a vote of thanks on the occasion of webinar on protein engineering. First of all, I am thankful to our provost, Dr. Anil Tomar sir, for providing an opportunity and giving permission to organize the webinar. I would like to express our profound gratitude to our guest and speaker, Professor C. Ratna Prabha Madam, who is Professor and Head Department of Biochemistry, MS University, Baroda. She has accepted our invitation and enlightened the webinar by sharing her experience and knowledge about protein engineering. The webinar would help us in understanding molecular mechanism of ubiquitin dependent and independent mechanism of protein degradation. I earnestly thank to all the participants from across the nation who have attended the webinar. At last, I would like to congratulate Dr. Sandesh Chiba and his team of Department of Life Science for successfully conducting the webinar. So at last, thank you very much to one and all.
Thank you.
Thank you, madam. Sir, your